Dusk settles gently over the wetland, and with it comes a subtle shift in the balance of power. The air cools, humidity thickens, and the boundary between land and water becomes indistinct. This is not an empty hour, but a transition window, one that countless organisms have evolved to exploit. Among them is a creature so small it often escapes notice until it has already succeeded. The mosquito does not roar into the scene or announce its presence with color or size. It enters quietly, carried on fragile wings that beat faster than the eye can follow, navigating by scent, heat, and the faint chemical signatures released by living bodies. In this narrow window between day and night, the mosquito becomes one of the most efficient biological transport systems ever produced by evolution. From a distance, the act appears trivial: a brief landing, a pinprick sensation, a momentary itch. Yet beneath that minor interaction lies a highly specialized feeding process that functions as a direct biological shortcut between organisms. The mosquito does not merely feed on blood, it taps into the circulatory system itself, bypassing the skin's defenses and inserting foreign material directly into a living bloodstream. In ecological terms, this is not just feeding behavior. It is a bridge, a highway that pathogens use to leap from one host to the next with astonishing efficiency. The mosquito's mouthparts are not a single needle, but a bundled instrument, a set of elongated structures folded together into a proboscis. When the insect lands, it does not stab blindly. Sensory receptors at the tip probe the skin, detecting capillaries beneath the surface. Once a suitable vessel is located, the mosquito inserts several microscopic stylets, each with a distinct role. One pair anchors the tissue, another pair slices gently through skin layers, and a central tube searches for blood flow. This process is precise, deliberate, and refined by millions of years of selection. The mosquito is not breaking the skin through brute force, but slipping between layers with surgical efficiency. As soon as the stellets penetrate, saliva flows into the wound. This saliva is not incidental. It is chemically complex and essential to the feeding process. It contains anticoagulants that prevent blood from clotting, vasodilators that increase blood flow, and compounds that dull pain responses just long enough for the insect to feed undetected. In evolutionary terms, the saliva exists to solve a mechanical problem: how to extract blood quickly without triggering defensive reactions. Yet this same saliva creates an open door. Any virus, parasite, or microscopic organism residing within the mosquito's body now has a direct route into a new host. Unlike predators that consume tissue externally, the mosquito establishes an intimate connection. Blood flows from the host into the insect, while saliva flows from the insect into the host, creating a brief but profound exchange. For pathogens, this moment is everything. Skin is normally an effective barrier, keeping foreign organisms out. The mosquito renders that barrier irrelevant. It transforms a complex immune landscape into a simple transfer point, turning individual animals into nodes within a larger network of transmission. Inside the mosquito, pathogens do not drift passively. Many undergo active development within the insect's body. After entering the mosquito during a blood meal, viruses and parasites must survive digestion, evade immune responses, and migrate to the salivary glands. Only then can they be passed on. This journey is perilous, and many fail. But those that succeed gain access to an extraordinarily reliable delivery system. The mosquito becomes not just a carrier, but an incubator, a moving environment where pathogens adapt to life between hosts. The efficiency of this system is amplified by the mosquito's feeding behavior. Female mosquitoes require blood to produce eggs, tying reproduction directly to repeated host contact. Each feeding event is a new opportunity for transmission, and each successful egg-laying cycle increases mosquito populations, expanding the network even further. In warm, stagnant environments where water accumulates, breeding sites multiply rapidly. Larvae develop in shallow pools, ditches, and flooded ground, often completing their transformation in days. The result is a sudden bloom of adult mosquitoes, all driven by the same biological imperative to feed. This is why outbreaks often follow seasonal rains or temperature shifts. Environmental changes do not merely increase mosquito numbers, they synchronize feeding behavior across entire populations. As more mosquitoes seek blood at the same time, the density of contacts between hosts rises sharply. Pathogens that might otherwise fade out find new life in this crowded biological marketplace. Transmission accelerates not because the pathogens have changed, but because the delivery system has become more active. The mosquito's sensory world is finely tuned to exploit this opportunity. Carbon dioxide plumes from breath guide it from a distance, while body heat and skin odors refine its approach. Even subtle differences in microbial communities on human skin can influence attraction, making some individuals more likely targets than others. From the mosquito's perspective, a host is not a person or an animal, but a map of chemical signals, each one narrowing the search until a suitable feeding site is found. This precision reduces wasted effort and increases feeding success, further strengthening the efficiency of transmission. Once feeding is complete, the mosquito departs unnoticed, leaving behind a microscopic trail of consequences. The immune system responds to the saliva, producing inflammation and itching. But if a pathogen has been introduced, the response may already be too late. Viruses can begin replicating within host cells almost immediately, while parasites may enter the bloodstream and disperse to specific tissues. The mosquito's role ends in seconds, yet the biological cascade it initiates can unfold over days, weeks, or even years. From an ecological perspective, mosquitoes occupy an uncomfortable position. They are prey for birds, fish, amphibians, and other insects, forming an integral part of food webs. Their larvae filter organic material from water, contributing to nutrient cycling in aquatic systems. Yet their feeding behavior places them at the center of some of the most significant disease dynamics on the planet. This dual role is not a contradiction, but a reminder that evolution optimizes for survival, not for harmony with human health. The mosquito does not intend to spread disease. It follows a narrow evolutionary script, one that prioritizes reproduction through blood feeding. Pathogens exploit this script, adapting themselves to fit the mosquito's life cycle and physiology. Over time, this partnership becomes finely tuned, producing transmission routes so efficient that a single bite can connect distant ecosystems, linking wildlife reservoirs, domestic animals, and human populations into a shared biological circuit. In this way, the mosquito represents the cleanest outbreak highway in nature. No wounds, no prolonged contact, no visible exchange. Just a brief landing, a silent injection, and a departure into the dark. The simplicity of the act hides its power. By piercing skin and injecting saliva, the mosquito collapses the distance between organisms, turning individual bodies into stepping stones for microscopic travelers. In the vast complexity of ecological interactions, few pathways are as direct, as efficient, or as consequential as the one traced by this fragile insect on beating wings. The forest floor of Madagascar is rarely still. Beneath the canopy, where sunlight fractures into shifting patches and humidity clings to every surface, decay is not a failure of life, but one of its most productive states. Leaves rot into soil, fallen fruit collapses under its own sugar-rich weight, and animal waste dissolves into a microbial soup that feeds the next generation of growth. In this layered world of breakdown and renewal, one of the most successful travelers moves without urgency or elegance. The Madagascar hissing cockroach advances methodically through this terrain, its body low to the ground, its movements steady, its presence almost architectural against the forest debris. It does not hunt, sting, or inject. It simply walks, and in doing so, it becomes one of nature's most effective mechanical carriers of contamination. Unlike the mosquito, whose danger lies in penetration, the cockroach operates entirely on the surface. Its biology is built for contact, persistence, and exposure. Every part of its body is designed to withstand environments that would disable or kill more delicate insects. Thick, waxy cuticle plates protect it from desiccation and chemical damage, while spined legs provide traction across slick organic matter. These legs are not smooth structures, but complex tools, lined with microscopic hairs and hooks that grip uneven surfaces. As the cockroach moves through decomposing material, feces, carcasses, and fungal growth, these structures collect what the environment offers: Bacteria adhere to the cuticle. Spores lodge between joints. Microbial films coat the feet like invisible paint. In the dense leaf litter of Madagascar, waste is everywhere, even when it is not obvious. Animal droppings sink into soil. Fruit ferments where it falls, and dead insects dissolve into nutrient-rich residue. The cockroach does not avoid these materials. It is drawn to them. Rot is food, and food is opportunity. As it feeds, it pushes its body into crevices where moisture and microbes concentrate. Each step presses its legs into surfaces teeming with bacteria that thrive on decay. There is no need for the microbes to survive inside the insect. They do not have to bypass immune systems or adapt to internal chemistry. They simply cling. What makes this form of transmission so effective is its simplicity. Mechanical transfer does not require biological compatibility. The cockroach does not need to be infected for it to be dangerous. It does not need to host a pathogen or allow it to reproduce. All it must do is move. As it travels from decomposing matter to fresh surfaces, it creates a physical bridge between environments that would otherwise remain separate. In human terms, this is not a needle-piercing skin, but a contaminated surface touched repeatedly by unknowing hands. The danger lies not in invasion, but in accumulation. The cockroach's body plan enhances this role. Its flattened shape allows it to squeeze into narrow spaces, increasing the range of surfaces it contacts. Cracks in bark, gaps beneath stones, and the undersides of fallen logs all become transit points. Each contact is an exchange. Microorganisms picked up in one location are deposited in another, smeared across new substrates by the friction of movement. In environments where food preparation or storage occurs, this behavior becomes especially consequential. A cockroach emerging from waste does not announce its origin. It leaves behind no visible mark, only microscopic traces that can persist long after the insect has moved on. The Madagascar hissing cockroach, despite its size and visibility, exemplifies this process rather than escaping it. Its large body increases surface area, providing more real estate for microbes to cling to. The hiss, produced by expelling air through spiracles, may startle predators, but it does nothing to alter the insect's ecological role. The true influence of the cockroach is silent, unfolding at a scale far below human perception. Bacteria transferred from one surface to another do not cause immediate dramatic effects. Instead, they build risk gradually, contaminating food, tools, and living spaces through repeated contact. From an evolutionary perspective, the cockroach's success is rooted in tolerance. It thrives where others fail because it does not rely on cleanliness or specialization. Its digestive system can process a wide range of organic material, including substances rich in microbes. While many bacteria are destroyed during digestion, others pass through the gut or remain on the exterior, unaffected. The insect's immune system is tuned to manage constant exposure rather than eliminate it entirely. This balance allows the cockroach to coexist with microbial communities that would overwhelm less resilient organisms. Predators play a role in shaping this dynamic. Birds, reptiles, and small mammals feed on cockroaches, keeping populations in check and recycling nutrients. Yet predation does not eliminate the insect's influence. Even a short lifespan filled with movement is enough to spread contamination widely. The cockroach does not need to be abundant to be effective. Its persistence, its ability to survive disturbance, and its tendency to revisit the same spaces repeatedly amplify its impact over time. The environments that favor cockroaches are often those where waste accumulates faster than it can be removed. In natural ecosystems, this is a normal and necessary condition. In human-altered spaces, it becomes a vulnerability. Food remnants, moisture, and shelter create microhabitats that mirror the forest floor, inviting cockroaches to move freely between waste and nourishment. Each journey retraces the same mechanical pathway, reinforcing patterns of contamination that are difficult to interrupt without changing the environment itself. What makes this mode of transmission particularly insidious is its invisibility. There is no bite to notice, no immediate reaction to signal danger. The cockroach leaves behind a smear rather than a wound, and the consequences emerge later, when bacteria introduced to a new surface find conditions suitable for growth. This delay disconnects cause and effect, allowing the insect to operate unchecked. The threat is not the cockroach itself, but the microbial history written across its body. In the broader ecological context, this behavior is not malicious. It is simply the result of an organism optimized for survival in decaying environments. The cockroach is a cleaner of sorts, breaking down organic waste and redistributing nutrients. The same traits that make it valuable in natural systems make it problematic in shared spaces with humans. Evolution does not distinguish between helpful and harmful outcomes. It selects for efficiency, and the cockroach is efficient at moving through the world. Seen this way, the Madagascar cockroach becomes a living surface, a mobile platform that connects places through touch alone. Every step it takes carries the memory of where it has been. In a landscape shaped by decay, that memory is harmless, even essential. In environments where cleanliness matters, it becomes a liability. The danger is not in what the cockroach does deliberately, but in what it cannot avoid doing. Wherever it walks, it leaves a trace, and wherever it leaves a trace, life follows. Under the open sky of a grassland, where wind presses waves into tall stems and large mammals move like slow weather systems across the horizon, the most consequential events often happen after the giants have passed. A fresh pile of dung lands heavily on the ground, steaming slightly as it meets cooler air, already alive with bacteria, parasites, and microscopic organisms that thrived inside another body only moments before. To the untrained eye, it is waste, an endpoint. To the dung beetle, it is a resource dense with energy, information, and opportunity. This is not merely food. It is raw biological material, and the beetle is one of the planet's most effective engineers for redistributing it. The arrival of dung is a signal that travels quickly through the ecosystem. Volatile chemicals released by fresh feces spread through the air, forming invisible trails that dung beetles can detect from surprising distances. Within minutes, the ground may host a flurry of activity as beetles converge, each drawn by the promise of nourishment and reproduction. Their bodies are compact and powerful, shaped by selection for strength rather than speed. Thick forelegs act like shovels, while ridged exoskeletons provide leverage against dense, sticky material. In these moments, the beetle's world narrows to texture and resistance, to the physics of pushing and lifting a substance far heavier than itself. Dung is among the richest microbial environments on land. It contains bacteria from the gut, parasite eggs shed during digestion, and spores from organisms that exploit nutrient-rich conditions. Left exposed, this material would remain on the surface, its biological contents slowly dispersing through wind, water, and scavengers. The dung beetle changes that trajectory. With deliberate effort, it cuts, shapes, and moves portions of feces away from the original pile. Some species tunnel directly beneath it, while others form nearly perfect spheres and roll them across the ground, navigating by celestial cues and landscape gradients. Each movement is a relocation event, transferring a dense package of microbes from one location to another. The act of rolling is not chaotic. It is precise and purposeful, shaped by evolutionary pressure to minimize competition and predation. As the beetle pushes its dung ball away from the source, it creates distance between itself and rivals, but it also stretches the microbial footprint of the original deposit across the landscape. Every rotation presses bacteria and parasite eggs into the soil surface, leaving traces along the path. When the beetle finally buries the dung, the relocation becomes vertical as well as horizontal. Material that once sat exposed is now carried below ground, inserted into darker, cooler layers of soil where conditions differ dramatically. This burial process has profound consequences. Soil is not uniform, it is stratified, with oxygen levels, moisture, and microbial communities changing with depth. By dragging dung underground, beetles introduce surface microbes into zones they might not otherwise reach. Some organisms perish, unable to survive the new conditions. Others persist, finding refuge from ultraviolet radiation and desiccation. Parasite eggs, in particular, benefit from this transport. Shielded from surface hazards, they can remain viable longer, waiting for the chance to encounter a suitable host. The beetle does not create pathogens, but it reshapes their geography. From a distance, this looks like cleanup, and in many respects it is. Dung beetles reduce surface waste, recycle nutrients, and improve soil structure. Plants benefit from increased nitrogen availability, and grazing animals encounter cleaner pastures. Yet beneath this ecological service lies a quieter redistribution of life at microscopic scales. The outbreak is no longer confined to bodies. It spreads into the ground itself, mapped by the tunnels and burial chambers of insects that never interact directly with hosts again. The beetle's physiology supports this role in subtle ways. Its digestive system can process dung efficiently, extracting nutrients while passing many microbes unharmed. Its cuticle, like that of other beetles, resists abrasion and chemical exposure, allowing it to operate in environments saturated with bacteria. Even its life cycle reinforces microbial movement. Eggs laid in buried dung hatch into larvae that feed within these subterranean chambers, further breaking down material and mixing it with surrounding soil. Over time, what was once a concentrated source of microbes becomes diffuse, spread through multiple layers and locations. Predators circle this process as well. Birds watch for beetles rolling their prize across open ground, timing strikes to snatch both insect and cargo. Mammals disturb soil while foraging, redistributing buried material once more. Rain infiltrates tunnels, carrying dissolved nutrients and microbes deeper still. The beetle's initial action sets off a cascade of secondary movements, each one expanding the reach of what began as a single deposit. This is where the concept of outbreak becomes ecological rather than individual. Pathogens and parasites do not need to move directly from host to host to persist. They can occupy landscapes, cycling through soil, insects, and intermediate organisms before returning to a body. Dung beetles act as vectors in space rather than flesh, reshaping where microbes live and how long they remain viable. In doing so, they influence infection risks indirectly, altering exposure patterns for grazing animals that later feed in the same area. Evolution has not shaped dung beetles to manage disease. Their behaviors exist to secure food and reproductive success. Yet these same behaviors demonstrate how deeply interconnected biological systems are. A beetle rolling a ball across a savanna participates in nutrient cycling, soil aeration, population control, and microbial redistribution all at once. The elegance of the system lies in its indifference. There is no intention, only consequence layered upon consequence. At night, when temperatures drop and many predators retreat, beetles continue their work under starlight. Some species orient using the Milky Way, rolling dung in straight lines guided by celestial patterns older than humanity. The image is cinematic, but the impact is grounded and physical. With each successful burial, the beetle locks organic matter into the soil, where it will feed plants, microbes, and unseen organisms for months or years. The outbreak does not explode in a single moment. It settles, diffuses, and waits. In this way, dung beetles reveal a different dimension of biological risk. Transmission does not always require contact between living bodies. Sometimes it unfolds through landscape engineering, through the quiet labor of insects that rearrange the world beneath our feet. The beetle never pierces skin or smears surfaces. It moves the ground itself, turning soil into an archive of past lives and potential futures. The outbreak here is written into the land. In the humid forests of Central and South America, the ground itself seems to move. Along roots, across fallen branches, and over the uneven skin of the forest floor, narrow highways pulse with life. Leafcutter ants flow along these routes in endless columns, each individual carrying a fragment of green, far larger than its own head. From a distance, the scene appears agricultural, even pastoral, as if the forest were being quietly harvested. But this is not foraging in the simple sense. What these ants are transporting is not food in its final form, but raw material for one of the most complex biological systems built by any non-human organism. A living fungus farm, whose survival depends on strict disease control. Leafcutter ants do not eat leaves. The plant matter they collect is carried underground, where it is chewed into pulp and used to cultivate a specific fungus that has co-evolved with the ants for millions of years. This fungus is their primary food source, providing nutrients unavailable in raw vegetation. The relationship is obligate on both sides. The fungus cannot survive without the ants, and the ants cannot survive without the fungus. What emerges from this partnership is something closer to a city than a nest, complete with specialized workers, waste management systems, and biosecurity protocols that rival those of human societies. Every foraging trail is a potential vulnerability. Leaves collected from the forest canopy carry more than chlorophyll. Their surfaces host spores, bacteria, and fungal competitors, invisible to the ants but potentially lethal to their crop. As ants cut and transport leaf fragments, they also move entire microbial communities from one environment to another. The trail itself becomes a corridor for contamination, linking distant plants, soil patches, and underground chambers into a single biological network. A single contaminated leaf introduced into the garden can seed an infection capable of overrunning the entire fungal crop. The danger is not hypothetical. Parasitic fungi exist that specialize in attacking the ants' cultivated fungus. These invaders grow faster, spread aggressively, and can collapse a colony if left unchecked. Unlike predators that remove individual ants, microbial threats target the core of the system. They do not need to kill workers directly. They only need to compromise the garden. The outbreak in this case is infrastructural. It spreads through shared material, shared air, and shared labor, turning the very efficiency of the colony into a liability. The ants respond with an arsenal shaped by evolutionary arms races that have played out far longer than human history. Sanitation begins on the trail. Workers groom themselves and each other constantly, scraping spores and debris from their bodies before entering sensitive areas. Contaminated material is often diverted to waste chambers located far from the fungal garden, creating physical separation between clean and dirty zones. These waste piles are not random dumps. They are deliberately placed and maintained, reducing the chance that harmful microbes drift back into the heart of the colony. At the center of this defense is chemistry. Leafcutter ants produce antimicrobial compounds from glands in their bodies, applying them directly to the fungal crop and to their own cuticles. These chemicals suppress the growth of unwanted microbes without harming the cultivated fungus. In addition, many species maintain symbiotic bacteria on their exoskeletons. These bacteria secrete antibiotics that target specific fungal pathogens, creating a living pharmaceutical layer that travels wherever the ant goes. Each worker becomes both a farmer and a medic, carrying treatment with it as it moves through the colony. This constant balancing act shapes every aspect of leafcutter behavior. Foraging routes are not chosen solely for efficiency, but also for safety. Trails may shift to avoid heavily contaminated areas, and certain plant species are rejected if they consistently introduce harmful microbes. Ants assess leaves not just by texture or chemistry, but by microbial risk, a sensory evaluation that unfolds without awareness, but with remarkable accuracy. The result is a dynamic map of the forest, constantly updated by experience and collective memory. Predators lurk along these trails, drawn by the abundance of moving prey. Birds, reptiles, and arthropods snatch ants from the columns, thinning their numbers. Yet predation rarely threatens the colony as a whole. Disease does. A successful pathogen can spread faster than any predator can hunt, exploiting shared spaces and repeated behaviors. This asymmetry explains why leafcutter ants invest so heavily in prevention rather than response. Once the garden is compromised, recovery is difficult and sometimes impossible. Inside the nest, the fungal chambers are carefully regulated environments. Temperature, humidity, and airflow are maintained within narrow ranges, optimal for fungal growth and hostile to many competitors. Ants adjust ventilation by opening or sealing tunnels, responding to subtle changes in conditions. This environmental control adds another layer of defense, turning the nest itself into a selective filter that favors the desired organism while excluding others. The ants are not merely reacting to disease. They are shaping the conditions under which disease can or cannot exist. The scale of this operation is staggering. A mature leafcutter colony can process tons of vegetation each year, sustaining a fungal biomass that outweighs the ants themselves many times over. With that scale comes risk. The more material that moves through the system, the more opportunities there are for contamination. Yet leafcutter ants persist, generation after generation, because their defenses evolve alongside the threats they face. Each outbreak that wipes out a colony leaves behind a lesson encoded in the genes of survivors. From an ecological perspective, leafcutter ants influence entire landscapes. Their foraging alters plant communities, their nests aerate soil, and their waste enriches the forest floor. At the same time, their disease management strategies limit the spread of certain microbes, shaping microbial communities in ways that ripple outward. The outbreak is never confined to the nest. It is negotiated continuously between ants, fungus, microbes, and environment. What makes this system so compelling is its familiarity. The challenges leafcutter ants face mirror those of human societies managing dense populations and shared infrastructure. Movement brings prosperity and risk. Connection accelerates growth and exposure. The difference lies in timescale and intention. The ants do not understand disease, yet they have evolved solutions that are elegant, effective, and deeply integrated into their way of life. In the dim light of the forest floor, as green fragments march steadily toward underground chambers, a quiet war unfolds. Chemistry meets infection. Behavior counters invasion. The outcome determines whether a colony thrives or collapses. The leafcutter ant does not merely survive in a world full of microbes. It confronts them head-on, transforming the outbreak from an existential threat into a manageable constant. In doing so, it reveals that some of the most sophisticated responses to disease are not planned, but grown, shaped slowly by evolution into systems that work because they must. Morning breaks slowly over a flowering landscape, not with silence, but with vibration. As the sun warms petals and leaves, the air begins to hum, first faintly, and then with growing intensity, as honeybees lift from their hives and fan outward into the surrounding terrain. From above, their movement resembles a living circulation system, thousands of small bodies tracing overlapping routes between nests and nectar sources. This traffic is not random. It is structured, repetitive, and dense, and in that density lies both the extraordinary success of the honeybee and one of its greatest vulnerabilities. Bees are not designed to pierce skin or smear waste. Their role in disease movement emerges from something far more fundamental: social life at scale. A honeybee colony is a superorganism, a tightly integrated collective in which individuals function more like cells than independent animals. Workers touch constantly, exchanging food mouth to mouth, grooming one another, and sharing chemical signals that coordinate behavior. Inside the hive, space is limited and movement is continuous. Bees crawl over one another as they tend brood, process nectar, and regulate temperature. This constant contact creates ideal conditions for the rapid spread of pathogens that do not need to survive outside the host for long. Viruses, bacteria, and parasites exploit the very behaviors that allow the colony to function. The journey begins outside on flowers. Each bloom visited by a bee is not just a source of nectar and pollen, but a meeting point where individuals from multiple colonies intersect. A single flower can host dozens of visitors over the course of a morning, each one leaving behind microscopic traces of where it has been. Pathogens shed onto floral surfaces can persist long enough to be picked up by the next forager. The flower becomes a shared resource, and with sharing comes exchange. This is not a dramatic invasion, but a quiet transfer, repeated thousands of times across a landscape. Foraging routes amplify this effect. Honeybees show strong site fidelity, returning to the same patches of flowers again and again. When resources are abundant, traffic intensifies. Bees from different hives converge on the same stands of blooming plants, brushing past one another in mid-air and landing on the same petals in rapid succession. The repeated contact does not require direct interaction between bees. The environment itself becomes an intermediary, holding and passing along pathogens adapted to survive these brief exposures. Once back at the hive, the real acceleration begins. Nectar collected in the field is not consumed immediately. It is passed from bee to bee through trophallaxis, a process that allows the colony to distribute food efficiently. Each exchange is intimate, involving direct contact between mouthparts and the transfer of liquid that may carry viral particles. Pollen, packed into pellets on the hind legs, is deposited into cells where it will be processed and consumed by larvae and adults alike. If pathogens have entered this flow, they gain access to every layer of the colony. Crowding intensifies during periods of growth. In spring and early summer, colonies expand rapidly, filling every available cell with brood. Workers cluster tightly to maintain optimal temperatures for developing larvae, generating heat through muscle activity and distributing it through constant movement. Thermoregulation, one of the colony's most impressive achievements, comes at a cost. Close contact, warm conditions, and high humidity create an environment where microbes can thrive. What benefits the young bees also benefits the organisms that exploit them. Parasites add another dimension to this system. Certain mites attach to bees and move with them between individuals and across life stages. These parasites feed on bodily fluids and weaken their hosts, but their greater impact lies in their ability to act as amplifiers. By creating wounds and suppressing immune responses, they make it easier for viruses to spread and replicate. A single infested bee can introduce a cascade of infections into a hive, not through aggression or invasion, but through routine social interaction. Drifting behavior extends the network beyond a single colony. Foragers sometimes enter the wrong hive, especially in dense apiaries or natural aggregations where nests are close together. This is not an error from the colony's perspective, but a consequence of navigation based on landmarks and scent, which can overlap in crowded environments. When a bee drifts, it carries with it the microbial signature of its home hive. The result is a bridge between colonies, allowing diseases to move through populations with surprising speed. Predators and environmental stressors influence this dynamic indirectly. When resources are scarce or habitats are fragmented, bees are forced to travel farther and concentrate more intensely on limited food sources. This increases contact rates and stress, both of which weaken immune defenses. In these moments, the social system that normally buffers the colony against individual loss becomes a conduit for collective vulnerability. The outbreak is not an external event, but an emergent property of connectivity under pressure. Yet honeybees are not passive in the face of these threats. They have evolved behaviors that mitigate disease spread, even if they cannot eliminate it. Workers groom themselves and others, removing parasites and debris. Infected individuals may be isolated or expelled, reducing transmission within the hive. Propolis, a resin collected from plants, is used to seal cracks and coat interior surfaces, creating antimicrobial barriers that limit microbial growth. These measures do not stop all pathogens, but they slow their progress, buying time for the colony to adapt or recover. The balance is delicate. High traffic and shared resources are essential for the efficiency of the colony, enabling rapid exploitation of food and coordinated responses to environmental change. The same features make the system fragile. Unlike solitary insects, bees cannot simply avoid contact. Their strength lies in numbers and cooperation, and those strengths come with inherent risk. Evolution has shaped their immune responses to operate at the colony level as much as the individual level, distributing the cost of infection across many bodies rather than allowing it to overwhelm one. In the broader ecosystem, this pattern has consequences. Diseases that move efficiently among bees can influence pollination dynamics, affecting plant reproduction and the animals that depend on those plants. The movement of pathogens through bee populations is intertwined with the movement of pollen through landscapes, linking health and productivity in ways that are easy to overlook. The outbreak here is not confined to a species. It ripples outward, subtly altering the fabric of ecological relationships. Standing near a hive at midday, the hum is constant and immersive. Bees arrive and depart in steady streams, each one a small participant in a vast collective enterprise. Their world is built on trust in proximity, on the assumption that contact is beneficial. Most of the time, that assumption holds. But when pathogens enter the system, they exploit the same channels that carry food and information. The dirty travel way of the honeybee is not dirt in the conventional sense. It is traffic. It is touch. It is the unavoidable consequence of living together at scale in a world full of invisible life. In the quiet margins of forests, gardens, and wooden structures left exposed to weather, another kind of city exists, one carved rather than built. Carpenter bees do not gather in hives or cultivate fields of flowers underground. They excavate. With powerful mandibles and a persistence shaped by evolution, they bore into wood, transforming beams, branches, and fence posts into long, smooth tunnels that run parallel to the grain. From the outside, these entrances look insignificant, little more than round punctures shadowed by sawdust. Inside, however, they open into dense residential blocks where biology accumulates layer by layer, generation by generation. Wood is not sterile. Especially when exposed to moisture, it becomes a living substrate, colonized by fungi, bacteria, and microscopic arthropods that thrive in darkness and dampness. Carpenter bees choose these materials not because they are clean, but because they are workable. Softened wood yields to their jaws, allowing tunnels to be extended year after year. The decision to nest here places the bees in constant contact with microbial films coating the tunnel walls, with mites that hitchhike through narrow corridors, and with spores that drift invisibly through the enclosed air. This is not a one-time exposure. It is a permanent condition of life inside wood. A single tunnel may host multiple brood cells, each separated by partitions of chewed wood pulp. Within these cells, larvae develop in close proximity to the materials that once supported previous generations. Old nests are not abandoned lightly. Carpenter bees often reuse existing tunnels, expanding them or subdividing them to accommodate new offspring. This reuse creates continuity. What lived here before leaves traces behind, not just structural, but biological. Microbes established in the wood persist, waiting for new hosts to arrive. The nesting cycle intensifies this effect. Adult females provision each brood cell with pollen and nectar before sealing it off. These resources are gathered from the outside world, carrying environmental microbes from flowers and soil. Once deposited, they become enclosed meals in warm, humid chambers, conditions ideal for microbial growth. If contaminants are present, they can proliferate alongside developing larvae, creating early life exposure that shapes immune responses and survival outcomes. The outbreak here is not explosive. It is slow, intimate, and persistent. Carpenter bees are solitary in behavior, but communal in architecture. Multiple females may nest in the same wooden structure, their tunnels running side by side like apartments in a block. Walls between tunnels are thin, sometimes separated by only a narrow layer of wood. Fungi do not respect these boundaries. As moisture fluctuates with weather, fungal networks can spread laterally through the grain, connecting spaces that appear isolated. Microbial communities migrate quietly, crossing from one tunnel to the next without any bee needing to move. Mites exploit this layout with particular efficiency. These tiny arthropods attach to adult bees as they move between tunnels and foraging sites. Some are relatively benign, feeding on debris. Others carry pathogens or weaken their hosts by draining resources. When a bee enters a new or reused tunnel, mites disembark, introducing whatever they carry into fresh spaces. Over time, a single contaminated tunnel can seed an entire structure, its influence spreading through shared walls and repeated visits. Predators rarely penetrate these wooden refuges directly. Birds may attack adults in flight, and parasitic insects may lay eggs near entrances, but the interior remains a protected environment. This protection, however, comes at a cost. Reduced airflow means moisture lingers. Temperature fluctuations are dampened, creating stable microclimates where fungi flourish. Carpenter bees regulate conditions only indirectly, through tunnel placement and depth, not through active ventilation. The result is a habitat that favors long-term microbial persistence. Generational overlap compounds the issue. In some species, offspring emerge into tunnels still bearing the biological signatures of their parents. Even when adults do not interact directly with their young, they share the same carved space, separated by time rather than distance. Microbes adapted to this environment benefit from continuity, encountering familiar conditions again and again. The wooden apartment block becomes an archive of past occupancy, its walls recording a history invisible to the naked eye. From an evolutionary perspective, this strategy is a trade. Nesting in wood offers protection from weather and many predators, increasing reproductive success. The risk of microbial accumulation is managed through resilience rather than avoidance. Carpenter bees possess immune systems capable of handling constant low-level exposure. They groom themselves, removing debris and mites where possible, but they cannot sterilize their environment. Instead, selection favors individuals that tolerate these conditions, passing that tolerance to their offspring. Environmental change alters this balance. Increased moisture from shifting climates or human irrigation can accelerate fungal growth within wooden structures. Human-built environments provide abundant nesting material, but often lack the natural decay cycles that limit microbial buildup in forests. Old beams remain in place, reused year after year, allowing biological loads to accumulate beyond what the bees evolved to manage. In these settings, the hidden outbreak intensifies, affecting survival rates and population dynamics. The ecological consequences extend outward. Carpenter bees are important pollinators, especially for plants requiring buzz pollination. Their health influences flowering success and plant diversity. When nesting environments become compromised, reproductive output declines, subtly altering pollination patterns across landscapes. The outbreak inside wood thus echoes into open air, connecting dark tunnels to sunlit flowers. Seen in cross-section, a carpenter bee nest resembles an orderly corridor system, cleanly carved and deceptively simple. But simplicity in structure masks complexity in interaction. Wood, moisture, microbes, mites, and insects form a layered system where movement is limited but influence is not. Each reused tunnel increases efficiency and risk simultaneously, reinforcing patterns that favor persistence over purity. There is a quiet irony in this arrangement. The carpenter bee, often viewed as a destroyer of wood, is in fact a tenant navigating an already living material. The wood is not passive. It hosts life that responds to excavation, colonizing new surfaces as they are exposed. The bee reshapes the habitat, and the habitat reshapes the bee. Their relationship is not clean or controlled. It is negotiated continuously in darkness. Inside these wooden apartment blocks, the outbreak does not announce itself. It does not surge through populations in dramatic waves. It lingers, passed along through reused spaces and inherited environments. The danger is not contact with outsiders, but familiarity, the comfort of returning to the same safe place again and again. In the world of carpenter bees, infection travels not on wings or trails, but through walls and time, hidden in the grain of the homes they carve. Heat hangs in the air above a late summer clearing, thick with the smell of ripe fruit and drying grass. Somewhere nearby, a carcass lies half hidden in the brush, already claimed by beetles and flies. From above, a wasp drops into the scene with sharp intent, its wings snapping briefly before folding as it lands. This insect does not specialize in one food source or one role. It hunts, scavenges, drinks, and samples whatever the landscape offers. That versatility is its strength, and it is also the reason the wasp becomes one of the most efficient mechanical couriers in the ecosystem, carrying invisible passengers between places that would otherwise remain biologically separate. Wasps move fast, not just in flight, but in decision-making. A patch of exposed meat, a fallen fig split open by birds, a spill of sugary liquid on a rock, all are assessed in seconds. The wasp lands, feeds briefly, then lifts off again, often traveling only a short distance before touching down somewhere new. Each stop adds another layer to what it carries. The mouthparts pick up residue. The feet press into surfaces rich with bacteria. Fine hairs on the legs and body trap particles too small to see, holding them just long enough to be deposited elsewhere. Unlike insects that specialize in clean nectar or sealed blood meals, wasps live in the overlap between decay and freshness. They tear protein from carcasses, chew insects into pulp, and then pivot immediately to fruit or human food sources without pause. This rapid switching creates high contact routes across different microbial worlds. Bacteria adapted to decomposing tissue suddenly find themselves on a grape skin or a picnic table. Microbes from soil and animal waste hitch rides to flowers, water sources, and nest entrances. The wasp does not discriminate. It connects. The mechanics are simple. Wasps feed using mandibles designed for cutting and tearing. These structures are robust and textured, ideal for gripping prey and scraping surfaces. As they work through soft tissue or fermenting fruit, fluids coat their mouthparts. When the wasp lifts off and lands again, traces remain. Feet play an equally important role. Each leg ends in pads and claws that provide traction across uneven terrain. These pads adhere lightly to surfaces, picking up films of moisture and the microorganisms suspended within them. The wasp cleans itself, but not continuously and not completely. Grooming is part of the wasp's daily routine. Individuals scrape their legs against their bodies, wiping debris away and redistributing it rather than eliminating it. This behavior reduces parasite loads and clears sensory structures, improving survival. It also shifts microbes from one body part to another, sometimes into places where they are better protected from desiccation or exposure. Grooming is maintenance, not sterilization. It lowers risk for the insect without breaking the chain of transfer. Venom adds an interesting complication. Wasps evolved venom primarily for hunting and defense, a complex cocktail that immobilizes prey and deters predators. Some components of this venom have antimicrobial properties, capable of inhibiting certain bacteria. When prey is stung and processed, microbial loads can be reduced locally. This gives the impression of cleanliness, a brief chemical reset at the point of capture. But venom does not coat the wasp's entire body, nor does it neutralize everything encountered. Its effects are limited in space and time, insufficient to counteract a lifestyle built on constant contact with diverse substrates. The wasp nest itself becomes a hub in this network. Workers return carrying food, prey fragments, and environmental debris. Inside the nest, larvae are fed through repeated mouth-to-mouth contact, each transfer an opportunity for microbes to spread. The nest environment is warm, humid, and densely populated, conditions that favor microbial persistence. Some pathogens struggle here and fade. Others adapt, using the predictable flow of resources and bodies to move efficiently through the colony. Predators interact with this system indirectly. Birds capture wasps mid-air or at feeding sites, removing individuals but also dispersing fragments of nests and prey. Spiders trap wasps in webs, creating new points where microbes from the wasp's body meet those of the web and surrounding vegetation. Even failed predation attempts contribute to redistribution, as stung or injured wasps land unexpectedly, touching surfaces they would not normally visit. Environmental context matters. In urban and agricultural settings, wasps exploit human waste streams with ease. Open trash, exposed food, and sugary residues provide abundant resources. Here, the contact routes intensify. A wasp that feeds on discarded meat can be on a piece of fruit moments later, then at a nest built under a roof beam, carrying microbial communities across artificial and natural boundaries. The speed of movement collapses distance, making separation meaningless. Evolution favors this behavior because it works. The wasp's generalist strategy allows it to thrive in changing environments, exploiting whatever resources appear. The cost of microbial exposure is managed through robust immune responses and short generation times rather than avoidance. Wasps do not attempt to keep clean in the human sense. They accept constant contamination as the price of flexibility. Over evolutionary time, this tolerance has proven more reliable than specialization. There is a subtle irony in their reputation. Wasps are often seen as aggressive and dangerous because of their sting, yet their more significant ecological impact lies in what they move rather than what they inject. The sting is obvious and memorable. The mechanical transfer is quiet and persistent. A brief visit leaves no mark, only a redistribution of microscopic life that continues long after the wasp has gone. At a landscape scale, wasps contribute to nutrient cycling by breaking down prey and carrion, accelerating decomposition. In doing so, they participate in the same microbial economy they help spread. They are both consumers and couriers, embedded in feedback loops that connect death to growth. The outbreak here is not an aberration. It is part of the system's function, a consequence of organisms interacting across boundaries that are invisible at small scales. Watching a wasp move through a summer afternoon reveals this pattern clearly. It never stays long. It samples, departs, and returns somewhere else, repeating the cycle until dusk. Each movement is efficient, purposeful, and loaded with consequence. The venom may neutralize a prey item. Grooming may reduce personal risk. But the lifestyle itself ensures contact, and contact ensures transfer. In the end, the wasp's role as a dirty courier is not a flaw but a feature of its design. It thrives in complexity by embracing it, moving between clean and unclean, living and dead, cultivated and wild without pause. The routes it creates are short, fast and crowded, perfect pathways for whatever can cling to legs and mouthparts long enough to make the journey. The outbreak does not need intention. It only needs motion, and few insects move through so many worlds in so little time. As morning light filters through the edge of a tropical forest, the first thing that appears is not the spider, but the structure it has left behind. Suspended between branches, glowing faintly with a metallic sheen, the web stretches outward like a lens, tuned to the movements of the air. Threads catch sunlight and bend it, revealing geometry so precise it appears engineered. This is the domain of the golden silk orb weaver, a spider that does not roam widely in search of prey but instead reshapes space itself, turning empty air into a checkpoint where traffic must pass. The web is not passive. It is a biological filter, placed deliberately along flight paths insects use as they move between feeding grounds, breeding sites, and waste-rich environments. Near ponds, over forest clearings, along the edges of human settlements, the orb weaver positions its trap, where movement is dense and unavoidable. Flies rising from carcasses, mosquitoes emerging from water, beetles departing dung pats, and wasps scavenging scraps all converge unknowingly on the same invisible barrier. By the time they reach the web, they are already carrying the microbial histories of where they have been. Insects rarely arrive clean. A fly lifting off from decomposing flesh carries bacteria on its legs and mouthparts. A mosquito leaving a stagnant pool brushes against algal films and microbial blooms. A beetle crawling out of dung is coated with spores and parasite eggs too small to register as weight. The web intercepts all of them equally. It does not discriminate by species or origin. Anything that collides becomes part of a concentrated exchange, pulled abruptly from motion into stillness. The silk itself plays an active role in this process. Golden silk orb weaver threads are strong, elastic, and slightly adhesive, designed to absorb the kinetic energy of flying insects without breaking. When prey strikes, it becomes wrapped in a matrix of fibers that press against every surface of its body. Legs, wings, eyes, and mouthparts all make contact at once. Whatever microbes were clinging to the insect are now smeared across silk that will remain exposed to the environment for hours or days. From the spider's perspective, this is efficiency. The web gathers energy-rich prey into a single location, allowing the spider to remain relatively still, conserving resources. From a microbial perspective, it is convergence. Pathogens from many sources are funneled into one point in space, layered onto the same structure repeatedly. The web becomes an archive of traffic, each strand holding traces of multiple journeys. The spider itself adds another layer. When it approaches captured prey, it bites and injects venom that immobilizes and begins external digestion. This venom breaks down tissues, releasing fluids that spread across both prey and silk. Some microbes are destroyed in this process, others diluted, others released from the insect's body into the surrounding fibers. The spider then feeds, pulling nutrients inward, while remnants remain behind. The web is cleaned selectively, not entirely. Damaged sections may be replaced, but large portions persist, carrying forward whatever they have collected. Environmental conditions shape how long this biological load remains active. In humid air, microbes persist longer on silk, protected from desiccation. In shaded forest edges, ultraviolet radiation is limited, allowing bacteria and spores to survive hours or days. Rain washes some material away, redistributing it onto leaves and soil below. In this way, the web does not just intercept traffic, it redirects it, concentrating microbes and then releasing them back into the ecosystem in pulses. Predators and scavengers interact with this system as well. Birds steal trapped insects, tearing prey free and carrying fragments elsewhere. Ants climb the web's support lines, harvesting remains and dragging them back to their nests. Each interaction spreads what the web has gathered, extending the influence of a single spider far beyond the radius of its trap. The orb weaver does not move much, but its effects travel outward through others. The spider's placement choices reveal an understanding shaped by selection. Webs are rebuilt frequently, but rarely randomly. Sites with consistent insect flow are reused, sometimes for weeks. These are often locations where different ecological streams intersect. Water meeting land, forest meeting clearing, human structures meeting vegetation. At these boundaries, insects from multiple habitats cross paths. The web turns these crossings into choke points, compressing diversity of movement into a single plane. This makes the orb weaver not just a predator, but a collector. It gathers biological material from across the landscape, condensing it into silk and prey remains. The outbreak, in this context, is spatial rather than social. Microbes that would normally remain dispersed across air, water, and soil are brought together repeatedly in one suspended net. The web becomes a microbial crossroads, a place where organisms from unrelated environments briefly coexist. The spider's own adaptations allow it to tolerate this exposure. Its cuticle resists microbial invasion, and grooming behaviors remove debris from its legs and body. Venom and digestive enzymes reduce some microbial loads before ingestion. Yet the spider does not sterilize its surroundings. It relies on resilience, not cleanliness. The silk itself is produced continuously, replaced as it degrades, allowing the system to reset gradually, rather than abruptly. At night, the web becomes nearly invisible, but its function continues. Nocturnal insects collide with threads they cannot see, adding new layers to the accumulation. By dawn, the web holds a record of the night's movement, each capture marking a connection between distant places. The spider waits, sensing vibrations, responding only when energy arrives. It is a patient node in a restless network. In broader ecological terms, the golden silk orb weaver plays a stabilizing role. By removing large numbers of flying insects, it regulates populations that might otherwise explode. At the same time, it influences how microbes move through the system, concentrating and redistributing them in ways that subtly shape exposure patterns for other organisms. The web is both barrier and conduit, stopping motion while enabling transfer. There is a quiet symmetry here. Insects move through the world carrying invisible cargo. The web intercepts them, collects that cargo, and then releases it again through decay, predation, and weather. Nothing is truly contained. It is only delayed, rerouted, reshaped. The outbreak does not originate with the spider, but the spider determines where it pauses. Seen against the light, the golden silk web looks delicate, almost ornamental. In reality, it is a dense interface between worlds, a place where garbage meets nectar, where ponds meet trees, where death meets growth. The spider at its center is not just waiting for food. It is managing flow, turning chaotic movement into ordered capture. In doing so, it reveals that some of the most important pathways in nature are not roads or trails or bites, but threads stretched quietly across the air, catching whatever passes through. The stillness is deliberate. In tall grass warmed by the late sun, a praying mantis holds its position with a precision that feels unnatural, as if the insect were carved from the stem it clings to. Its triangular head remains motionless, forelegs folded in a posture that suggests patience rather than aggression. Around it, the ecosystem moves continuously. Flies lift from damp soil, Wasps zigzag between fallen fruit and flowering weeds. Moths flutter in irregular patterns as they search for nectar. The mantis does not chase this motion. It waits for it to come close enough to end. Predators like the mantis occupy a unique position in biological networks. They do not merely consume prey. They interrupt movement. Where insects act as carriers, transferring microbes across surfaces, habitats and hosts, ambush predators function as biological dead ends. An insect caught in mantis forelegs completes its journey there. Whatever it carried stops moving. In this way, predation shapes not just population size, but the flow of microscopic life through the environment. The praying mantis is exquisitely adapted to this role. Its body blends seamlessly into vegetation, aided by coloration that matches leaves, bark, or flowers depending on species. Some mantises sway gently, mimicking plant movement in the wind, a form of dynamic camouflage that fools both prey and predators. Large compound eyes provide depth perception uncommon among insects, allowing precise distance judgment critical for a strike that must succeed on the first attempt. This sensory accuracy ensures efficiency. Fewer failed attacks mean fewer wounded insects escaping back into the environment. When the strike comes, it is sudden and final. The mantis's raptorial forelegs snap forward, spines locking around wings and thorax. Escape is rare. The prey is immobilized and brought toward powerful mandibles that begin consumption immediately. For insects weakened by disease, parasites, or age, this moment often arrives sooner. Reduced reaction speed, erratic flight, or diminished strength makes them easier targets. Predators tend to remove the vulnerable first, a pattern observed across ecosystems. In doing so, mantises reduce the number of compromised insects continuing to move through shared spaces. This selective pressure has important consequences. Insects carrying heavy parasite loads or suffering from infection often behave differently, flying lower, resting longer, or failing to evade danger. By removing these individuals, predators can lower the density of potential carriers without ever encountering the pathogens directly. The outbreak route narrows. Fewer insects travel between waste, water, and food sources. The environment becomes quieter in ways that are invisible but measurable. The mantis's influence extends beyond the moment of capture. By occupying strategic ambush sites near flowering plants, water edges, and insect corridors, it inserts itself into high traffic zones. These are the same routes used by insects that have visited dung, carcasses, stagnant pools, or fermenting fruit. Each successful kill represents not just energy gained, but movement stopped. The mantis does not cleanse the environment actively, yet its presence alters the statistical flow of contact events. However, nature rarely offers solutions without trade-offs. The mantis is not immune to the biological consequences of its diet. When it consumes prey, it also consumes whatever that prey carries internally. Parasites that infect insects often rely on predators to complete their life cycles, using the predator's gut as the next environment in which to develop. Ingesting infected prey exposes the mantis to these organisms, turning the dead end into a transition point rather than a terminus. Some parasites survive digestion, passing through tissues or insisting within the predator. Others exploit the predator's physiology more aggressively, reducing lifespan or reproductive success. This cost is unevenly distributed. A mantis that feeds heavily on contaminated prey faces greater risk than one hunting cleaner insects. The cleanup job, while beneficial at the ecosystem level, exacts a toll at the individual level. Evolution balances this cost by favoring behaviors and immune responses that tolerate low-level exposure, rather than eliminate it entirely. The mantis's digestive system reflects this balance. Strong enzymes break down prey rapidly, limiting the window during which parasites can act. Gut environments are hostile to many microbes, yet not all. Some persist, adapted to survive in predators precisely because predation is common. The mantis becomes part of a longer chain, not a final barrier. The outbreak route bends rather than ends. Predators themselves are prey. Birds, reptiles, and mammals hunt mantises, especially larger species. When a mantis carrying parasites is eaten, those organisms gain another opportunity to move upward through the food web. In this way, disease dynamics extend vertically, linking insects, predators, and higher consumers. The mantis reduces horizontal spread between insects, while participating in vertical transfer through trophic levels. Control and risk coexist in the same body. Environmental context shapes the mantis's role. In diverse habitats with many predators, no single species dominates removal. In simplified environments, mantises can become primary regulators, their absence or presence having outsized effects. Agricultural landscapes often benefit from mantis predation, as pests weakened by pathogens are removed before populations surge. Yet these same environments may concentrate contaminants, increasing the risks predators face. The effectiveness of the dead end depends on the balance of pressure and exposure. There is also a temporal dimension. Mantises are sight and weight hunters, active during daylight when insect traffic peaks. Their stillness conserves energy, allowing long periods between meals. During these intervals, insects move freely and routes remain open. Predation does not eliminate movement entirely. It punctuates it. Each capture is a pause in an otherwise continuous flow, enough to matter over time, but never enough to sterilize a system. This partial control is precisely what ecosystems favor. Total elimination would collapse food webs. Instead, predators like mantises apply constant, uneven pressure, shaping behavior and survival without halting interaction. Insects that evade predation tend to be stronger, faster, or less burdened by disease. Over generations, this pressure can reduce the prevalence of certain pathogens indirectly, selecting for hosts that manage infection more effectively. Yet the mantis itself must navigate these pressures. As it ages, exposure accumulates. Parasites ingested earlier may influence later performance, slowing strikes or weakening grip strength. A once perfect ambush predator becomes more vulnerable, more likely to be caught by birds or reptiles. In this way, the cost of cleanup is deferred rather than avoided. The mantis pays eventually, contributing its own body back into the system. Seen in profile against the sky, the praying mantis embodies a paradox. It is a symbol of stillness in a world of movement, a predator that halts journeys while quietly participating in others. It reduces risk by removing carriers, yet accepts risk by consuming them. Its presence improves the health of the system, even as it shortens its own odds. Nature's sanitation is never free. In the broader story of outbreaks, the mantis reminds us that control often comes from removal rather than prevention. No chemical barrier, no grooming behavior, no engineered structure can match the simplicity of stopping movement entirely. Predators do this constantly, invisibly, without intention. They thin the lines, break the chains, and shorten the routes along which disease travels. But they do not escape the consequences. Every dead end has an entry cost. The mantis absorbs it with each meal, trading personal safety for ecological function. In tall grass and flowering fields, this trade plays out daily, a quiet exchange between risk and regulation. The outbreak routes narrow, then widen again, shaped not by design, but by hunger, patience, and the sudden snap of spined forelegs closing around a life that will not travel any farther. In the soft light of a temperate morning, leaves glisten with moisture that has not yet lifted into the air. Stems bend slightly under the weight of dew, and the green surface of the plant world becomes a continuous landscape, rather than a collection of separate organisms. This is where the ladybug lives. Bright, rounded, and instantly recognizable, it moves slowly at first, its domed body catching light as it climbs from leaf to leaf. To most observers, the ladybug appears gentle, even ornamental, a harmless presence among aphids and flowers. But beneath that familiar shell is one of the most persistent travelers in the plant world, an insect whose entire lifestyle is built on constant contact with the very surfaces where microbes thrive. Plants are not clean. Their leaves and stems host dense communities of bacteria, fungal spores, yeasts, and microscopic organisms adapted to life on waxy cuticles and in thin films of moisture. Every leaf is a habitat, every stem a corridor. Wind deposits spores from distant fields. Rain splashes microbes upward from soil. Insects feed, defecate and die on plant surfaces, adding their own biological signatures. This phyllosphere, the living skin of plants, is one of the most extensive microbial environments on land. The ladybug does not pass over it. It walks through it. Unlike insects that move in bursts between distant points, ladybugs travel methodically. They crawl. Their feet grip the surface of leaves, press into veins, and trace along stems with deliberate precision. Each step brings their tarsal pads into direct contact with microbial films invisible to the naked eye. Fine hairs and adhesive structures allow the insect to cling even to smooth surfaces, and these same structures collect whatever the plant surface offers. Bacteria, spores and particles adhere easily, held just long enough to be carried onward. The ladybug's foraging behavior intensifies this process. Aphids, their primary prey, cluster on new growth where plant tissues are tender and nutrient-rich. These areas are also hotspots for microbial activity, as sap flow and insect damage create entry points for fungi and bacteria. As ladybugs hunt, they move repeatedly between infested plants, climbing up stems, crossing leaf edges, and descending again. The journey is continuous, a chain of contact events linking one plant to the next. Feeding does not interrupt this movement. When a ladybug consumes aphids, it anchors itself firmly to the plant surface, bracing against leaves as it works. Fluids from prey can spill onto the plant, mixing insect and plant-associated microbes. When feeding is complete, the ladybug resumes its crawl, carrying traces of both environments forward. There is no pause for cleaning beyond brief grooming motions that redistribute material rather than remove it entirely. Predators shape this behavior indirectly. Birds and other insects hunt ladybugs, especially when their bright coloration fails to deter inexperienced attackers. To avoid danger, ladybugs drop from plants or take flight, landing elsewhere moments later. Each landing initiates a new sequence of contact. Flight compresses distance, turning what might have been a slow crawl into a sudden leap across the plant network. The microbial cargo remains with the insect, bridging gaps that wind and water alone might not cross. Seasonal changes expand the scale of this movement. As temperatures fall, ladybugs migrate in large numbers, sometimes traveling kilometers to overwintering sites. Along the way, they stop repeatedly on vegetation, resting and feeding as needed. These pauses are brief but frequent, enough to create a chain of plant-to-plant transfer across landscapes. Even during migration, the ladybug remains a walker at heart, touching down often and making contact wherever it lands. The mechanics of attachment are central to this role. Ladybug feet are equipped with pads that secrete a thin layer of fluid, increasing adhesion. This secretion interacts with the surface chemistry of leaves, forming temporary bonds strong enough to support the insect's weight. In doing so, it also creates a medium in which microbes can survive briefly, protected from immediate desiccation. When the foot lifts, residues remain on both sides. Transfer does not require intention, only motion. Plants respond to this traffic in complex ways. Some microbes carried by insects are harmless or even beneficial, contributing to plant defenses or nutrient cycles. Others are pathogens, capable of causing disease if introduced to vulnerable tissues. The ladybug does not distinguish between them. It moves whatever adheres, spreading diversity as readily as danger. In agricultural systems, this can influence how plant diseases move through crops, especially when insect populations are high and vegetation is dense. The ladybug's own biology encourages tolerance rather than avoidance. Its immune system manages constant exposure to environmental microbes without eliminating them entirely. Chemical defenses, including alkaloids that make the insect unpalatable to predators, do not sterilize its exterior. Bright coloration warns birds, but does nothing to deter fungi or bacteria. The ladybug survives by accepting its place in a crowded microbial world, not by escaping it. Reproduction reinforces these patterns. Eggs are laid on leaves near aphid colonies, placing the next generation immediately into the same contact-rich environment. Larvae hatch and begin crawling at once, their elongated bodies tracing the same paths as adults, but with even greater surface area in contact with plant tissues. Before they ever take flight, they have already moved across multiple plants, participating fully in the mechanical redistribution of microscopic life. Environmental moisture amplifies the effect. Morning dew and light rain create continuous films across leaves and stems, allowing microbes to survive longer outside their original niches. Under these conditions, the ladybug's movement becomes especially effective. Spores picked up on one leaf remain viable long enough to be deposited on the next. The outbreak route does not require wounds or injections. It follows the contours of plants themselves. There is a quiet contrast here between appearance and function. The ladybug's rounded shape and bright colors evoke harmlessness, even friendliness. Yet its ecological role is relentless. It is almost always in contact with something, always moving, always bridging small gaps. Where larger insects leap or fly over surfaces, the ladybug traces them carefully, step by step, ensuring transfer through persistence rather than speed. This is why plant-to-plant transmission can be so effective. Leaves often touch or nearly touch, forming networks that insects can traverse without ever leaving the plant layer. Ladybugs exploit these connections naturally, turning vegetation into a continuous highway. In this space, microbes do not need vectors that bite or sting. They only need carriers that walk. At a broader scale, ladybugs contribute to balance by controlling aphid populations, reducing the spread of some plant diseases indirectly by limiting pest damage. Yet at the same time, they facilitate movement of other organisms simply by being present. Control and transfer coexist, reflecting the complexity of ecological roles that cannot be reduced to single functions. Standing back and watching a ladybug climb a stem reveals this truth clearly. Its journey is slow, almost meditative, yet it covers ground relentlessly. Each step presses into the living skin of plants, collecting and releasing unseen passengers. The outbreak does not announce itself. It rides quietly on feet, passed along through routine movement rather than dramatic events. In the end, the ladybug teaches a final lesson about how disease and disruption travel through nature. Not all outbreaks need bites, wounds, or direct invasion. Some spread through touch alone, carried patiently across surfaces by organisms that never leave the layer where microbes already live. The ladybug, harmless in appearance and gentle in motion, embodies this reality. In a world where life is connected by contact, even the smallest steps can carry consequences far beyond what they seem.